Bismillahirrahmanirrahim. Our topic for today is rod, Shigella. It affects those children who are under 10 years. Now, the mode of transmission is by 4Fs. The 4F dictates that it can be caused by either flies, fingers, food, or feces. Now, the foodborne breakout, if compared to the waterborne breakout, it is 2 to 1 ratio, means food breakout is comparatively more. Now, this organism infectivity is quite dangerous. Even a small concentration of this can cause a lethal effect. Now, whenever this organism is ingested, this affects the distal ileum and the colon. Now, what this organism do is it penetrates into the distal ileum mucosa. By reaching the mucosa, it enters the submucosal layer, where it will cause a cascade of inflammatory process in which a lot of neutrophilic aggregation will occur. There will be a lot of macrophages present in that respective area. So predominant, this would be a point of severe form of inflammation. If inflammation is going on, the upper endothelium, what happens is there will be an ulceration formation. Because of this ulceration formation, before that there will be leakage of fluid, initially there will be watery diarrhea, but as inflammation slowly progresses around this respective area, there will be slowly bloody diarrhea, hence the name bacillary dysentery. Now, always remember, the amount of concentration of organisms in this type of dysentery is comparatively very low in comparison to that of salmonella, and this is an only human disease, and this organism is a non-lactose, as similar to that of salmonella. But the differentiating feature is that this organism does not produce H2 gas or hydrogen sulfide gas in a TSI medium similar to that of salmonella. Got ingested by the ways of by fingers, by having contaminated food through the flies, or by orophycal routes such as contaminated feces which interacts with food. So now the incubation period of this organism within the human body is of 1 to 4 days. Now we know whenever it causes intestinal irritation, there will be inflammation, which will be associated with fever and abdominal cramps. Then we know that inflammation would result in leakage of fluid from the intestine, resulting in watery diarrhea, and finally, because of ulceration, there will be bloody diarrhea. Now, these symptoms resolve within 2 to 3 days. This will completely resolve, and antibiotics can shorten the course of this disease. Labs will again start from culturing this organism, and we will use McConkey agar, CONKEY, and EMB agar, because whenever we acquire a GIT rod, a gastroenteric rod, we always have to identify if it is a lactose fermented or not in order to eradicate E. coli from our D. So we know that there will be no fermentation. So we'll move next to TSI. On TSI, we will rule out our next organism, which is salmonella. So if no H2S gas is formed, we are clear that even our salmonella is being ruled out. Then for the confirmation of basically this organism, we will move towards slide agglutination. Now in our slide agglutination, there will be a slide agglutination of the respective antibodies, which will confirm the finding of the toxin. This is produced by this organism, which is known as Shigella toxin. This is the toxin which was excreted in the gastrointestinal tract, which result in inflammation and ulceration process. Now, next, after doing this, after confirmation of this organism, we can also do a methylene blue stain for fecal sampling. Methylene blue. Now, if in stool, which you can see here, if you can find neutrophilic invasion in this, we can confirm that the organism present was probably Shigella, Salmonella, or Campylobacter. If the neutrophils are not present in that respective stool, we can rule these out, and we can probably think that the next possible organism that could be inhabiting the intestine would be Vibrio cholera, E. coli, because it results in non-blood diarrhea, or C. perfringens. Now, moving towards the treatment of this is that we know that it is a self-limiting disease, but diarrhea is always accompanied with dehydration. So we need to give the patient lots of fluids and maintain electrolyte. By preventing imbalance, we can avoid a lot of complications. Electrolytes. And in mild cases, mostly no antibiotics are given, but in severe cases, we start off with ciprofloxacin. So this is only recommended for severe cases, which is a fluoroquinolone is a drug of choice. But recently, plasmid have there are a lot been genetic variation, so multi-drug resistance has occurred. So alternate drug, if there is resistance, we can start off the patient with, if we're suspecting that there will be multi-drug resistance because of the frequent breakouts, we can use trimethoprim. Trimethoprim, sulfamethoxazole. And accompanied with those drugs, we can also use antiperistaltic, such as metronidazole,